and welcome to The Scoop. Today we have an interesting duo who are going to take us behind the science, research, and technology of personalized glycemic control using the gut microbiome. This cutting-edge information that we're going to share today is based on 5 years of research conducted by two co-founders of Day 2, Professor Segal and Professor Elinav from the Weizmann Institute of Science in Israel. The research from this project was funded solely by the Weizmann Institute of Science, and upon its completion, the research technology was licensed to Day 2. This research has been recently published in the Journal of Nature, the Journal of Science, and the Journals of Cell and Cell Metabolism. And presenting this very interesting research today is Ms. Tali Raveh-Sadka and Chavi Kramer. Tali Raveh-Sadka is the Director of Research at Day 2. She's currently a postdoctoral fellow at the University of California, Berkeley, and secured her PhD from the Weizmann Institute of Science in Israel. Tali has worked under the direct supervision of Professor Eran Segal, one of the scientists behind the science, research, and technology of Day 2. Tali will be going through the research aspects of Day 2, and Chavi Kramer, the Senior Director Dietitian at Day 2, will break down the research that will enable practitioners to help their clients balance blood sugar levels with a personalized nutrition plan based on the Day 2 research and technology. Welcome, ladies, and thank you for joining us today.
Thank you, Shannon, for the introduction. Hi, everyone. As Shannon said, my name is Tali Raveh-Sadka, and I'm happy to be here today. So, as Shannon introduced, this technology is based on scientific research by Eran, Segal, and Elinav that you see here from the Weizmann Institute of Science, and this research is independently funded by the Weizmann Institute. And what I'm going to start by showing is a research published in the journal Cell. Nutrition scientists have struggled to find what is the best diet for humans. There's so many opinions: low fat, low carb, plant-based, high protein, paleo diet, but none seem to work for everybody. And as you know, we have changed and are continuing to change our recommendations over time, sometimes even reversing them. For example, eggs were once considered bad for cholesterol, but now we know that this is controversial at best. But for any given end goal, the question of what is the best diet is a scientific one. If diet A is better than diet B, then a clinical trial that compares the two diets on enough people for a desired outcome should show that definitively. Now, if there is such a thing as the best diet, then clearly we have not found it yet. If you live in the United States, there is more than 70% chance that you are either overweight, have diabetes, or have non-alcoholic fatty liver disease. There is overwhelming evidence that diet and lifestyle are major drivers for these conditions. So why is it that after so many research efforts, we still don't have an answer for the question of what is the best diet? Could it be because we've been asking the wrong questions? Asking what the best diet for humans is assumes that it depends only on the food and not on the person eating it. What if the differences in personal features such as genetics, lifestyle, and gut bacteria cause us to respond differently to food? Can these differences explain why some diets work for some people but not for others? What if our nutrition needs need to be personally tailored to our unique makeups? This is exactly the question that Professor Eran Segal and Dr. Elinav from the Weizmann Institute set out to study. To take a scientific approach to the question, they searched for a good metric of healthy nutrition that they could study. Most studies look at weight loss or risk for heart disease for following some diet. But the problem is that these are affected by many factors aside from diet, take many weeks to change, and in the end, they only give a single measure of success. And if it didn't work, well, then it's very hard to understand what excuse me. They decided to focus on blood glucose, and more precisely, changes in the postprandial glycemic response. These are the glycemic responses following meals. They chose that because spikes in glucose levels after meals directly affect fat storage and weight gain, and can make someone feel hungry and eat more. The spikes have been shown to be an independent risk factor for multiple diseases. Importantly, continuous glucose monitors can measure blood glucose every 5 minutes, allowing direct quantification of the effect of every single meal, as opposed to the more common practice of measuring the effect of an entire diet. Obviously, there are many factors beyond blood glucose levels that influence a healthy diet, but this is an important one which is believed to affect health in many ways. The Weizmann team connected 1,000 healthy participants to continuous glucose sensors and tracked their blood glucose levels continuously for a full week. During that week, participants logged everything they ate on a mobile app, and their exercise and sleep patterns were recorded with a Fitbit device. Participants also provided additional information such as age, weight, height, medical background, and completed food frequency questionnaires. They took blood tests and DNA sequencing of both their human genome and gut bacteria composition, using both 16S and full shotgun sequencing. The gut microbiome was probably the most novel factor they examined. Other studies they had done on the microbiome led them to ask whether the microbiome could also be important for explaining differences in meal glucose responses from the study group. What makes the microbiome even more exciting is its therapeutic potential. Unlike our genetics, we can change the microbiome to some extent, even by simple means such as changing what we eat. That offers a new tool for dietitians to use in medical nutrition therapy. So what did they find? When looking at averages, there were trends. For example, for most people in the study, more carbs in the meal generally increased the glucose response. More fat generally lowered the glucose response. But the key finding of the study was that for every trend they found, there were many people that had very different responses. When the same person ate the same meal on different days, the response was fairly similar. But when different people ate the same meal, the responses could be very different. For example, white bread had almost no effect on the blood sugar levels of some people, but in others, it elicited big spikes. And the same was true for every single food they tested, including rice, sushi, pizza, and even chocolate. For every food, some people had low responses, others had medium responses, and yet others had very high responses. It wasn't just about the food, it was also about the person eating it. So although averages and trends are informative, for any given individual, they may not mean much. And it wasn't just a matter of how well the body was handling sugar. Each person had specific foods that spiked their glucose levels. Many people even had opposite responses. For example, some people spiked for ice cream but not for rice, and others spiked for rice but not for ice cream. In fact, more people spiked for rice than for ice cream. Now, this may be shocking to practitioners who today rely on general dietary guidelines and advise people to eat more complex carbs such as brown rice. For most patients, that dietary advice in fact pushes them to develop faster the very disease that they're trying to prevent. The conclusion is that the diets that maintain normal blood sugar levels have to be personally tailored. And the best diet for all humans just doesn't exist. The scientists then asked if this variability across people was associated with any of the clinical or microbiome parameters that were collected. And indeed, many associations were found to blood parameters and other measurements. Interestingly, some associations with gut bacteria composition and specific bacterial metabolic pathways were enriched in people that had either high or low response to a particular food. The next question the scientists asked was how to predict each person's postprandial response to complex meals. Using a standard machine learning approach, a model was trained on data from 800 participants. Then the data was validated on a new set of 100 people whose data was not seen during model development stage. The algorithm that was developed predicts a person's personalized glucose response to just about any meal with high accuracy. As a final step, they wanted to see that the algorithm-based diet can indeed normalize blood sugar levels. New participants were recruited and profiled. Most of them had prediabetes. The algorithm was asked to design 2 diets for each person. One diet consisted of meals that were predicted by the algorithm to spike that person's glucose level, and the other consisted of meals that were predicted by the algorithm to induce a low glycemic response for that person. Each person then followed each diet for 1 week while connected to a continuous glucose monitor, and the microbiome was sequenced every day. By design, these diets were identical in their calories. It's also important to note that each person received a different personalized diet, and there were even foods that were on the low glycemic diet for some people and on the high glycemic diet of others. To show you that the diets are not the obvious ones you might think of, here they are for one of our participants. As you can see, each diet contains foods such as ice cream and chocolate that would not typically appear in standard diets. So take a moment here and think, which do you think was the high glycemic diet and which do you think was the low glycemic diet? So for this person, it turns out that the diet on the right with the ice cream was the one predicted to be the low glycemic diet. And how well did these diets work? What you'll see next is probably the most striking result in the study. So here are the continuous glucose levels of the participant during the diet that was predicted to yield high glycemic responses, where you can clearly see abnormally high glucose levels after meals. This indicates that this participant probably has prediabetes. But on the low glycemic diet, which includes that ice cream that we saw, and has the same number of calories as the second diet, the same participants had a fully normal postprandial glucose level without even a single spike across the entire week. And similar results were found in most participants. Both diets also induced several consistent changes in the microbiome of most participants. These changes were in line with previous studies that showed associations between certain bacteria and weight loss or glycemic control. Bacteria previously associated with beneficial effects increased in the predicted lower glycemic diet, and bacteria associated with harmful effects increased in the predicted higher glycemic diet. This is very intriguing because it suggests that in addition to normalizing blood sugar levels, the preferred diet induced beneficial effects that may persist even beyond the intervention week. This research was published in the journal Cell in November 2015, and it starts a new era in personalized nutrition, one in which we will move away from asking what is the best diet for humans and instead focus on the more appropriate question of what is the best diet for me. As a follow-up to the original study, in the beginning of 2016, the Weizmann scientists decided to dive deeper into bread. This study was published in Cell Metabolism in 2017. More than half of the world's population consumes bread every day, and it accounts for 10% of our caloric intake. Yet we know relatively little about its effects on metabolic parameters and our microbiome composition. There are many types of bread, and here it was decided to focus on 2 extremes. The first is white bread, made from refined wheat, in which the germ and bran of the wheat are removed, an industrial white bread that includes food preservatives and emulsifiers, and leavened with an industrial single yeast strain, was compared with a whole grain sourdough bread made from whole wheat flour and leavened with naturally occurring bacteria and yeast. The design was a randomized crossover trial with 20 participants. Some participants consumed the sourdough bread first for 1 week, followed by a 2-week washout period, and then 1 week of white bread consumption. The other 10 did exactly the same thing, but in opposite order. The study measured blood parameters and microbiome before and after each intervention week. So which bread was better? To their great surprise, using linear mixed models, which are widely used for analyzing crossover trials, they found no significant differences in the effect of the two breads induced on all parameters that were measured. Well, maybe bread just doesn't have any effect. They studied whether 1 week of intervention with bread of any type had any effect on metabolic and microbiome parameters. And quite surprisingly, they found that adding bread to your diet for just 1 week is sufficient to induce highly significant changes in several key metabolic parameters. Every dot in this plot represents the change, excuse me, the change from baseline of a specific participant, with the gray area indicating that the parameter went down. Some of these changes would be considered beneficial, such as reduction in LDL cholesterol, liver enzymes, and C-reactive proteins, but others would be considered adverse, such as the reduction in key minerals like iron, magnesium, and calcium. On the microbiome, though, they found that people generally maintained their overall microbiome species composition throughout the entire intervention. So in this CTOA plot, each dot is a sample, and ellipses represent samples from the same person before and after the white and sourdough interventions. And you can see that despite the significant dietary intervention that was done, microbiome composition remains person-specific throughout the entire trial. Overall consumption of bread has significant effects on key metabolic parameters, but there were no differences between the two bread types. So either the two breads really have the same effect, or, more interestingly, perhaps their effect is personalized and varies across people, such that when averaged across participants, you just don't see it. The bread type does not affect the overall average. So again, using postprandial glucose response as the metric, they indeed saw that some people responded more highly to sourdough and others more highly to white bread. In fact, you can see here where each dot represents a difference in the response to white and sourdough bread of a particular participant, that about half the participants had higher responses to white bread and half to sourdough bread. And this is very much in line to what we just saw in the cell study. So can we predict in advance which bread will be better for which participant? Using gut microbiome data alone, they again developed a machine learning model that could classify which bread induces higher glucose response with a good area under the ROC curve of 0.8. So how can a personalized nutrition plan be developed for each of us and the patients that we serve? First, a gut microbiome test kit from day 2 is used to identify the person's microbiome. Then, combined with a variety of personal parameters describing the person's lifestyle, diet habits, and general health, a personalized diet is developed. And the data is available through an app. Chavi, Day 2's on-staff dietitian, has created a video to review just that. Every day, she works with practitioners and patients alike to help them navigate the app and begin planning a balanced glycemic diet.
Thank you, Tali. So as Tali mentioned, I'm a dietitian at Day 2, and I also have my own private practice. I'm just going to start by going through the customer journey. So the customer goes online, purchases the Day 2 kit, fills out a short health questionnaire, then receives the kit in the post within a week, sends back the stool sample to our labs in Israel for DNA sequencing of the gut bacteria, and then receives his results back in the form of an app, which is available both on the mobile and the web. So I'm going to be going through the practical application of the web app, which is a little different to the mobile app, and how I see health practitioners using this in their practice when building meal plans. So you can see on the screen that we have 4 different categories when we're first going into the app. This is a test user called James Hobbs. And we're just going to start off by going into the first category, which is single food items. And I'm just going to click on an example over here of fruits. So the first thing that you notice at the top is the maximum daily allowance for James, which is 2,150 calories, and that's for weight maintenance. So if you're wanting to adjust that for your client to gain weight or to lose weight or a different kind of calculation, that's fine. This is just what the app suggests. And then we have a list of fruits with scores, which are personal scores for James, from 1 to 10, and as the score is higher, it means that that food will result in a less of a glucose spike for James. Now, the important thing to point out here is that the score can change. So if we add something to the food item or adjust the portion size, the glycemic response will be different. So let's do an example of a banana, and let's see if we can actually make this something possible for James to eat. So we're going to edit the meal. And usually the things that will help with the glycemic response, as Tali mentioned in the research, is fat or protein. And obviously that depends on the person. But in this example, we're going to add some walnuts to the banana and see if for James he actually responds positively to fat. So we'll select the walnuts from the list and choose 7 walnut halves. We're going to add that to the meal and see how this affects his score. So we can actually see that now it's possible for him to eat the banana. It's a really good snack for him. As a practitioner, you can save this meal for him for later use and suggest this for him to eat as a snack. Now, like I mentioned, this won't be the case with everyone. So I just want to show for demonstrative purposes a different test user that we have, Harold, the same example. And let's see if for him, the combination of the banana and the walnuts will also result in a good glycemic response. Now, we also see differences between, even if 2 foods have the same score, the effect of the fat or the protein will be different for each person. So we can see here again, banana is not good for Harold, but will the walnuts improve his score like it did with James? So we'll add the same amount as we did previously. We're going to add 7 halves. And we can see that the score is still not very good for Harold, so it's not a good option for him. So this is just an example of a simple meal of how we can actually improve scores in the app. I want to now show you, if you're going to build meal plans for your clients, how you can actually use this app when choosing meals. So firstly, we have pre-built meals in the app. We have a few 100,000 recipes and meals that you can choose with your client. You can also adjust those meals, so you can always open them up, look at the ingredients and tailor them to your client's preferences. Or you could build a meal from scratch based on your recommendations and based on what your client likes to eat. So we're going to do that over here, and we're going to build a meal of potato with some salmon, broccoli, and red wine and see how this works for James. So we'll start off with a baked potato, which, as we know, doesn't have such a great glycemic response in general. Let's see how it is for James, personally, and we can see that it's not so good. So now we're going to see what happens once we start adding all the other ingredients. So we'll start off by adding some broccoli. Now, vegetables don't normally help the score. They sometimes will leave the score the same, or they might reduce the score. So let's see what happens in this example. We can see the score went down. So it's obviously not something that you would want James to avoid, because we know that broccoli has benefits. So let's see if we add some salmon, if this can become a good meal for him. So it did help, but not significantly enough to make it a good option. So we're going to add a little bit more fat to this meal and see how this helps James. We'll add a tablespoon of oil, and the score jumped up. Now, finally, I just want to see what happens when we add some red wine, if this has an effect on the meal or not. So we'll add 1 glass of red Merlot. And you can see the score really improved in this case. So certainly as a practitioner, you might not be recommending red wine with every meal, but in this case, it's nice to see that it's a good option sometimes and can actually improve the score. So again, you can save this to James' favorites and go back to that for later use. So how do we put this all together when you're actually building a meal plan? So as I said, you can save the meals to favorites when you're building them. You can also choose meals from the meals category as well to save them to your favorites. And once you've come up together with your client with a few examples of breakfast, lunch, dinners, and snacks, you can then start to put that into your own meal plan template for your client. If it's based on calorie allowance, or if you don't work with calories, that's fine as well. We actually have a calorie counter that we're going to be adding to the app in the next week, which will also help you to use the app itself to save these meals. So by providing your client with a meal plan that combines your own approach together with the day 2 recommendations, you can really refine these recommendations to actually suit your client's needs. So thank you for your time today, and I'll pass you back to Tali.
So thank you, Chavi, and thank you everyone for joining the webinar. To close it, I would like to spend a brief moment to talk about where the research is heading in regards to the science and technology. Currently, we are pursuing clinical trials to validate the science completed at the Weizmann, as well as clinical trials in diabetes management to concretely study how the science and technology affect those with type 2 diabetes over the long run. The studies will focus on reductions in glucose variants and A1C levels. All of this will be used to build a case for FDA approval of the test kit and app for the reduction of A1C levels for those with type 2 diabetes. We will continue to build up the technology, making it more user-friendly when it comes to accessing the immense wealth of data powering the technology. We are currently working on building the app out further to include integrations into many other nutrition services and applications to help those applications and services better care for their end users. Additionally, we are building out our health practitioner program, providing a portal and system to enable dietitians and other practitioners to better manage their patients and access their patients' results, enabling them to make better meal plans, gain insights into their patients' gut microbiome, and ultimately provide better care. To conclude, Day 2 hopes to foster a new era in the growth of personalized medicine and nutritional care with the gut microbiome. We believe that once users understand that nutrition must be personally tailored, they will expect that any nutrition product or service will be personally tailored for them. As we help usher in this new era, we hope to give dietitians the science and the tools necessary to provide the level of personalized care to be expected, improving patient health and leading to a healthier society as a whole. Thank you very much, and now we'll have time for some questions and answers. Shannon?
All right, great. Thank you, ladies. Can you talk a little bit more? In the beginning, you talked about the testing that was done with rice and bread. Can you talk a little bit about how those results came about and maybe how the body responds differently for some people and not others?
So I'm not sure I'm following your question. So this is actually an observation. Since people were connected to continuous glucose monitors for a whole week, eating what they usually eat, but when you do that for a cohort of 1,000 people, you get many repetitions just because many people eat rice or bread or ice cream for that matter. So what the scientists at the Weizmann did was just to look at the responses to the same meals eaten by different participants. And what they saw is indeed that different people responded differently. In addition to that, there was also a set of standardized meals that were eaten at the same time by all participants and virtually handed to the participants to ensure that they were exactly the same. These were a set of breakfast meals. Some consisted, for example, those of bread with butter or bread without butter. And this was also used as some standardized way of measuring a response, the variability in responses across people. Did that answer your question, or should I clarify more?
Yeah, I think that answers my question. Let's see, we've got what happens if your client changes or aims to improve their gut microbiome via probiotics, fermented foods, etc.?
So right now, the science is not quite there yet, unfortunately. This is something we are actually working on actively, but at the moment, there is no good way of knowing how to direct your microbiome using probiotics, and there is no good enough scientific backing to being able to directly modify your microbiome in a beneficial way using these supplements. This is something we are researching actively, but this is not something that is currently part of our recommendations or part of what is included in the algorithm or the app, simply because there is not enough scientific backing for that at the moment.
Okay, great. Did you also look at weight gain or weight loss in the studies?
So not in these studies at all. We are looking at this internally now, using our own cohort, but this is very initial and still small study. So I hope we'll have something to share further down the line.
Okay, great. Can you talk a bit more about one's microbiome test results and how they dictate the diet, and are recommendations based on existing studies showing correlations between certain bacterial strains and food or glucose responses?
So first of all, about the test. So the test is using a fecal material test. We send out a collection tube for each of our participants that is later analyzed in our lab. What we perform is a metagenomic sequencing, that is a whole genome sequencing of all bacteria and all microbes present in the gut. We build a profile of species composition, as well as the abundance of different metabolic pathways in the sample for that participant. And this is fed into our algorithm, and the machine learning algorithm basically found patterns in all the data, not only the microbiome parameters. It looks simultaneously at all the different features, the BMI, the weight, the A1P levels, the microbiome features, looks at everything and tries to find pattern that it can generalize with regards to glycemic responses. And this is what our algorithm is based on. So we do not feed outside associations, published associations, to specific microbiome species when we make our predictions. Instead, this is a very complex machine learning algorithm based on gradient boosting trees, decision trees, and so it takes into account a lot of intercorrelations and interassociations between these features, and finds these patterns based on which it makes recommendations. And as I said, this algorithm was tested and cross-validated also, and validated on a different set of people. And we continually monitor that and keep on retraining the algorithm and testing it.
Okay, great. So I know that you tested several bread types. How many bread types and were tested, like carbohydrates, rather, like rice and various types of grains? You know, what does that look like? Like, how many different grains and breads were tested? And
so in the bread study that the second study that I went over, there were two types of bread that were tested: the white industrial type versus the sourdough bread. In the first, and this was the only thing that was tested in that study. In the first study that we went over, and that is actually the basis for the Day 2 technology, there was no, it was not aimed for specific or for a specific set of grains that was tested. Instead, as I said, people just ate their usual food. But when you take a group of 1,000 people, you get a fairly extensive sampling of all foods that people actually eat. So we have a very diverse set of grains that people ate, but not for, if there are some eccentric ones, then you would probably see fewer examples of that.
Okay.
But we've seen a very diverse set of foods.
Okay, cool. Did you also test like different types or qualities of grains, and did you test like dairy, different types of dairy, cow dairy, goat dairy, and whether different aspects of like farming, or if they were sustainably farmed or conventionally farmed? Did you do any of that kind of research in the project?
So no, not we haven't done any of these analyses. And to be honest, on the Israeli cohort that we've worked with, we didn't have access for that type of information. But as we are now expanding to a much more significant cohort in the United States, these are the sorts of questions we could start looking at.
Great. How do medications or antibiotics play in the microbiome? Were you able, when you tested these subjects, were there guidelines that restricted, supplementation, medication, the antibiotics, those types of medications in the study, or were they part of it?
Yes, so there were various exclusion criteria. For example, we prefer people to not provide us with a sample if they took antibiotics recently. We do not exclude people based on supplements or probiotics. And there are also various other exclusion criteria that were more aimed for the scientific study. It's less to make the cohort more controlled. As a company, we have a smaller set of restrictions because we do want to allow a larger set of people to have access to this technology. But I think that Chavi might have a more complete list of the restrictions we currently have in mind. Is that true, Chavi?
Yeah, so antibiotics is one of them. If someone has taken them within 3 months, then we say you need to wait. Also chemotherapy, within 5 years, we say you need to wait. Someone who's pregnant can't do the test, they'll need to wait 2 months. And people under 18, and anyone taking insulin. That's the exclusion criteria for the company at the moment. And just to add also, if anyone has any complex medical conditions, we encourage our customers to ask, and we check it, we look into it with our medical team, because obviously each condition is very, very specific. So we don't have a list of medical conditions, but people usually ask, and then we check and get back to them.
So were diabetes patients involved in this study?
Yes. Yes, not a very large percent of them, and now we have studies within Day 2 and also in the Weizmann Institute that are focusing specifically on diabetic and pre-diabetic populations. but there were a small but significant percent of both pre-diabetic and diabetic patients.
Okay, great.
And within the company, people with type 2 diabetes do it quite a lot. And just to add also, the study itself was validated in a study done by the Mayo Clinic. So it's also done on a US cohort as well to validate those findings.
An important addition. Thank you, Chavi.
Great, thank you for that. So if a client has gut dysbiosis or candida overgrowth, does that affect the results, or do you take that into consideration?
So this is taken into consideration the same way that all other microbiome features are taken into consideration. So if the algorithm was able to make out a pattern matching any kind of overgrowth or dysbiosis to a specific glycemic response, then it will definitely use it when making predictions.
And just also to add to Tali's point, it's also important to understand that the test itself is not a diagnostic test. We're not trying to diagnose dysbiosis or anything wrong with people's gut. We're using that information to predict glucose response, but we're not diagnosing anything.
Right, right. Are you able to monitor with the findings if the patient's response to the foods have changed, if they've improved once you've gotten the results and they're following more of the guidelines? Can you monitor improvement by
So at the moment, the way that we monitor improvement, or the way that our customers monitor improvement, is through blood tests. So checking their HbA1c after 3 months, checking their fasting glucose levels. Weight loss is another byproduct, even though we don't really say that our product is specifically for weight loss, but people do lose weight because of the connection between weight and insulin resistance. The other things, people will say they're feeling a lot more energetic, improvement in their exercise levels, overall, better mood, more focused. As a company, we do have, as Tali mentioned, some small internal studies where we're tracking things in a more formal way, like HbA1c and weight and other blood tests. But customers on their own, we usually tell them to check their blood tests and their weight and their energy levels. Cravings for sweet things as well as another one.
Okay, great. Have there been any studies in the U.S.?
You asked whether there's been studies in the U.S. I'm sorry, I couldn't hear.
Yeah, I know that the research has been done in Israel, but I'm curious if there have been studies in the U.S.
Right. Yes. So as Chavi mentioned, we are really in these days submitting 2 papers based on a study done in collaboration with the Mayo Clinic, which basically tried to revalidate the same study, the same self-study that was done in the Israeli population on an American cohort. And this is indeed what we found. We were able to validate it and see the same observations. First, that the responses are indeed variable across people, and also that our algorithm is able to provide a more accurate prediction of glycemic responses also on American clients. And so this will hopefully be published soon. We're just submitting it now.
So right now you're just working with the Mayo Clinic here in the States?
At the moment, we're working with the Mayo Clinic, but we are also establishing several other collaborations with different targeted populations, for example, diabetic patients. These will likely start soon, and we do have a very quickly increasing American cohort of clients that we hope to be able to see some significant improvements on them soon.
Yeah, that'll be interesting. I know I'm guessing, having not ever been to Israel, that the diet is quite different between cultures and regions.
It is. It is. This is exactly what we, yes, this is exactly what we are seeing in that Mayo study that we have, that I just mentioned. We do see the differences in eating patterns, in how much fiber is eaten. It's very much, and even in the microbiome composition, the changes that are expected as a result of eating less fiber and different kinds of food. We actually do see that in our data.
Oh, interesting that you can actually see that.
Yes, it is very interesting.
Right. Okay, so you do take into consideration the fiber content of the diet and the kind of the qualities of the foods. How can you update this with these, different countries of region? Are you able to update them with new foods or new quality of foods, since now we've got GMOs and conventional farming and the different types of agriculture and sustainable farming, etc.?
Right. So we have different food databases for countries, and we are continuously expanding and validating them using various taggings, like new sorts of taggings. For example, the type of diet, but this can also include the type of fiber. This is a lot of ongoing work that needs to be done in order to tag items in meaningful ways, and we're constantly looking for different sections to look at. Javi, do you have anything to add on that?
Yeah, I think something you mentioned earlier also, it is quite difficult when you're talking about things like agriculture and ways of cooking and that type of thing, as opposed to nutrients. It's a lot more difficult to measure. And in the original research, if you don't have all that information for each food item, it's harder to implement into the algorithm. So at the moment, it's mainly looking at nutrients rather than things like GMO or farming. But because we are expanding more and more, and because we're getting more US clients and different databases, hopefully this is something that can be implemented in the future as well.
Very interesting. Are you seeing like case studies in like how long have you been working with the Mayo Clinic, and are you testing? How many clients have you tested here in the States? Very interesting.
So in that study, we had around 330 participants. That study concluded. It spanned about a year and a half, and it is concluded now. But we have many more U.S. clients at the moment that are not a part of a specific study, and there will be more studies that will be kicking off soon, hopefully, with additional, as I said, more specific populations that we're targeting.
Excellent. Well, keep us in mind if you need study participants. We'd also like to know more about, can you tell us a little bit more about the practitioner health program and how that works?
Sure. So we have relationship both in Israel and in America with health practitioners. Whoever does want to partner with Day 2, we maybe, I don't know if Tred wants to jump in here a little bit to discuss it. Tred? No? Okay. So,
yeah, sorry.
Oh, yeah. Maybe you want to talk about the health practitioner?
Yeah, so basically what we do, it is the health practitioner program. It allows, we use the word practitioner pretty loosely, anything from endocrinologists to private practitioners to chiropractors, anything in between. It allows them to get Day 2 kits at the wholesale price and then resell them to their patients. So whether you want to, save those savings for yourself, pass them on to your patient, it does allow you to get the kits a little bit cheaper. And what we really like about that program is that we release the results directly to the practitioner. So you'll get the results before or when your patient does. That way you can go through the results and make sure there's nothing in there that might violate the care plan. Kind of like Chavi was mentioned earlier about serious medical conditions, because there are things that we aren't going to gather from day 2. So we really like it when we can get the practitioners involved to make sure that the results we're giving are used most effectively and most efficiently.
And NANP professional members have been authorized to use those kits, I understand from Josh.
Yes.
Great. And are those results, are they being used, or those samples, are they being used in any way for research or any type of clinical correlation, or how are they working within the states? And just as including that, you know, with the states being so huge and so diverse in eating philosophies and habits, it seems like that's going to give you just an incredible range of research to check out.
Yeah, so the problem is really not the lack of things to research at the moment, and we are really one of our plans is indeed to focus on the few different regions in the United States to get a better sampling of, and also different ethnicities, to get a better sampling of the U.S. population so that we can better match the nutritional needs of the population. Sorry, was there anything else you asked there?
No, I think that was it. Do we have any more questions from our attendees? We had a lot of questions today. And if there's no more questions, I just want to thank you all on your team for joining us. Oh, I've got a question here. Will you be attending the NANP conference? I don't know if any of you, Tread might be aware, but we have been in communication. They've got it on their calendar and they're looking to see if that's something that they will be able to attend. The researchers are in Israel, and several of the panelists today are from out of the country. So it's really at this point just depending on availability for different parts of the team. Some of the team are here and some of the team is abroad. And Tread, if you'd like to or are able to expand on that, please do.
Yeah, we definitely haven't finalized the decision. We'd like to be involved and be there and have a presence. But like you said, there's a lot of moving pieces, and they're including getting, you know, the principal scientists or CEO over from Israel in order to speak. But we will do everything we can to at least have a presence and meet some of your members.
Excellent. That would be wonderful. Okay, great. Thank you, team, day 2, for joining us. I think we're going to go ahead and close for now. This was a great informative talk, and we're really interested in what you're doing and are very interested in knowing how your research continues to progress and hopefully be a part of the great work that you have done and will be doing in the future. Is there any last closing comments you'd like to make before we end the webinar?
No, thank you so much for having us.
Okay, thank you for...
Yeah, thank you very much.
Thank you for your expertise and all of the work that you have done thus far. And again, we're going to be keeping our eye out on you guys and interested in the research you're doing, and very interested to see what you come up with here in the States. We've got your contact information there, SueMiller@day2.com. So if anybody has questions, please feel free to reach out to somebody in the team, and I'm sure they'll be able to direct you. So thank you, ladies and gentlemen, for joining us today. We really appreciate your time and your energy in this project.
Thank you.
Thank you, Shannon.
Okay.
Thank you.
All right, thank you. And this brings us to the end of our discussion. Thank you everyone for joining us and attending today's broadcast. We will be sending this out, so if you have questions for me or our panelists, please feel free to reach out and let us know what you thought of today's webinar. And also to remind you of next month on Thursday, September 13th, Mr. Reed Davis will be joining us with more empowering research on normal bone density scores and how they may not be enough. So please mark your calendars for Thursday, September 13th, and plan on joining us for another great presentation. And thanks for being with us on the call today. Have a great rest of your day.